Hello, everyone from Italy and Japan. Thank you for participating in today's workshop entitled Parkinson's Disease from Pathophysiology to Treatment. Parkinson's disease is an intractable disorder affecting more than 8 million people worldwide. We, Juntendo University, have been working on this disease from its birth more than a half century ago. All three department chairmen, Professor Narabayashi, Professor Mizuno, and Professor Hattori, have worked on Parkinson's disease, making Juntendo University one of the most prestigious places for clinical and research fields for Parkinson's disease. This workshop covers the recent findings which have unraveled the disease mechanism to pave the way for new treatment options. Professor Hattori will start with an overview of the history of the Parkinson's disease, highlighting major findings for this disease. Then, Dr. Hatano will present alpha-synuclein propagation theory, which is now considered an essential part of disease development. Dr. Saiki will explain recent advances in biomarkers, which enable us to detect the disease in the early stage. Finally, me, Dr. Tsunemi, will discuss exosomes, which will significantly contribute to the disease progression. Professor Hattori, please start your talk.
So thank you very much for kind introduction. So Professor Tsunemi, I will be talking about Parkinson's disease from pathology to treatment, especially history of Parkinson's disease, 200 years history. So since first essay by James Parkinson. So Parkinson's disease, chronic, progressive, neurodegenerative disorder, high frequency next to Alzheimer's disease, more than 10 million people worldwide are affected. Clinically characterized by akinasia or bradykinasia, essential for diagnosis of Parkinson's disease. In addition, resting tremor were deleted. So at least one of them are essential for diagnosis of Parkinson's disease. Pathological characterized by the loss of dopaminergic neuron in substantia nigra. But recently, so this like Lewy body formation could be observed in not only brain, but also other organisms, such as gut, or skin, or mandible. The remaining neuron contained Lewy body. Lewy body so consisting Lewy body consisting of alpha-synuclein protein. So this we call Lewy body and synuclein pathology. 95% of the patients are sporadic, but 5% of the patients are familial. This means hereditary Parkinson's disease could be also identified in the worldwide. Pharmacological and device-aided therapy dramatically improve the patient quality of life. So James Parkinson wrote an essay on the shaking palsy, levodopa as the first treatment for PD. So many discoveries. And alpha-synuclein is the first gene to cause Parkinson's disease. And the second causative gene, Parkin is the causative gene for young onset Parkinson's disease. And MIBD, this is unique or specific examination and for Parkinson's disease. And our group identified in addition to Parkinson, we have identified two genes, CHAD2 and prosapacin. So James Parkinson, born April 11th, 1755 in London. So in 1783, married Mary Dale. And in 1784, qualified as a surgeon, approved by the City of London Cooperation. And very interesting information. So James Parkinson had a chance to hear John Hunter lectured. John Hunter was talking about Parkinson's disease symptom. And such lecture provided him a good hint to write an essay on the shaken palsy. And in 1961, so levodopa as the first treatment for Parkinson's disease. This one, the video, L-DOPA treatment for patients with Parkinson's disease, 50 milligram levodopa intravenous injection before treatment of levodopa therapy. Steeple posture, reduce arm swing, small steps. Very slowness of gait after 50 milligram levodopa IV, dramatically improved by this treatment. Good activity, good gait, full swing, arm swing. One step, very good. So now this slide shows you development of Parkinson's disease treatment. Torihexiphenidil, and this video in 1961, the first trial for levodopa IV. And it's 1972, oral levodopa treatment has been started. Amantadine, bromocription. So 1979, so we had dopamine agonist treatment. And in 1980, levodopa plus DCI carbidopa or benseragide. And in 1989, so this is the only Japan and the USA, L-serol dopes, Paglar, Tarpexol, Cabergoline, Pramapexol, Lopinirol, and entacapone. Serotonin, the first medication of monoamine oxidase inhibitor. Zonisamide, this is the original anti-epileptic drug. But recently, zonisamide may have multi-potential agent and calcium ion, so reflux antagonist. And also pramipexole, long-acting dopamine agonist, apomorphine. Apomorphine could be used for off-stage as rescue treatment. Lochinocin so patch is different. So this is the adenosine A2A antagonist. Lasagirin, so this is the in Japan history of Parkinson's disease treatment. And the rasagirin second one is monoamine in B inhibitor. Sulfonamide also monoamine B inhibitor. Lopinirole patch, this is available in only Japan. Opicapone, this is a Compton inhibitor. And so James Parkinson mentioned, did not mention about familial Parkinson disease, but 1997, alpha-synuclein is the first gene to cause Parkinson disease. Second one, Parkin, is a causative gene. And CHD2 and prosapocin. Now, at least 25 genes, candidate genes for familial Parkinson disease. So this slide shows you PARK1 to PARK24. And recently, our group, this is the secret gene, PARK25 gene X, autosomal dominant. So PARK2, PARK22, and PARK24, three causative genes have been identified by our group. And very interesting, the yellow color shows you mitochondrial related gene. So faint green color shows you vesicular trafficking or synaptic related gene. So this is PARK2 and PARK22 and PARK24. And so not only oral medication, but also device-aided therapy has been developing so far. Among them, deep brain stimulation DBS, subthalamic nucleus or GPI, STN or GPI. And also very interesting, the MRBG cardiac scintigraphy, method of assessment of cardiovascular sympathetic function. So MRBG is a noradrenergic analog. So single photon emission computed tomography images were acquired at 15 minutes and 180 minute delay imaging after injection of MRBG. So this is the data, Parkinson disease, reduced uptake of MRBG. This is Parkinson disease, MSZ, essential tremor, and the control, and the vascular Parkinsonism. AB healthy control, AB healthy control, C stage 1, and the D so D is the Parkinson disease. And this is the axonal alpha-synuclein aggregate. Here are the centripetal degeneration of cardiac sympathetic nerve in Parkinson disease. Upper panel shows you tyrosine dehydrogenase, dopamine, and alpha-synuclein derivative. So this is the normal individual, DLB, and the Parkinson disease. Significant reduced staining for dopamine, hydrolase. So multiple system atrophy. So this is DBS, so deep brain stimulation. Courtesy by my younger colleague, Professor Umemura before DBS. So this is the switch off IPD. Now switch on. This patient showed dramatically improved after DBS, deep brain stimulation.
Yeah. Very dramatic. Sure, thanks a lot. Wow. Do that again. Did you jump up? Did you jump up?
Very soon, responsible
Pretty
for
cool.
DBS.
There he goes. Wow, I thought it would take longer. It's instantaneous. Just open both your hands like this. Focus back home.
So X-ray imaging of this patient head. So lead and IPD generated, pulse generated, and DBS. So year by year, IPD has been developing so far. Now one IPD, one IPD, okay, so as now. So this is scheme for DBS, pulse generated IPG, and that for diskinase GPI, globus pallidus internus, and usually we select SCN, subthalamic nucleus. So a lot of DBS for Parkinson's disease, long-term medication, disease progression, this initially phase we call honeymoon, and slowly progression. And then if patient 5 to 1, this is the criteria for DBS, more than 5 times intact of levodopa, and even though 5 times of levodopa, but such patient has more than 2 hours of time. So we have to consider DBS. After DBS, so good responsiveness, we call second honeymoon. So another aspect of DBS may have potential to reduce dose of levodopa. Approximately just half of them, like compared to before, so reduce levodopa. And then after second honeymoon, the slowly progression. So this is the randomized trial DBS for Parkinson's disease. So in New England Journal of Medicine, so this is a very good paper, 156 patient. So very good responsiveness. And then also very interesting data have been published. So many data have been published so far. Is Parkinson's disease a prion disorder? As you know, so mad cow disease is one of prion disorder. So prion protein that converted to normal prion protein, and then abnormal prion protein impressed upon converting to alpha pelix prion protein to beta sheet structure. The same story, alpha-synuclein also changing to beta sheet structure alpha-synuclein and fibril alpha-synuclein. And so subsequently, such alpha-synuclein fibril formed Lewy body. So recently, gut to brain or node to brain, the two dual hit hypothesis of propagation of a synuclein during Parkinson's disease. This is the Professor Black proposed this hypothesis. This hypothesis is fascinating, very interesting. So recently, gut to brain or olfactory valve to brain. But this is the only two pathways to brain. So my younger colleague, Dr. Tak, will be talk about discrepancy of dual hit hypothesis. But very interesting, transplanted dopaminergic neuron. So after embryonic neuron transplantation, but embryonic neuron transplantation, pathology findings shows you within neuron transplant in Parkinson's disease, you can see brown color shows you synuclein aggregation. So this data rewarded engrafted neurons in subject with Parkinson's disease suggests how to graft disease propagation. This is alpha-synuclein propagation story. Single inoculation alpha-synuclein fibrate enough to cause Parkinson's disease in mice. So this is the first paper in Science. Injection into abnormal synuclein. So this is the rapid and progressive rapid propagation. And finally, I will show IPS. IPS in medicine, induction of pluripotent stem cells from mouse embryonic and adult fibroblast cultured by defined factors. So Professor Shinya Yamanaka, Nobel laureate. Induction the IPS adult human fibroblast defined factors. This factor we call Yamanaka factors, 4 factors. And IPS-derived dopaminergic neuron improved only one case, a patient with Parkinson's disease. This paper published in 2020. So at 18 to 24 months after implantation, on and off, months since first implantation. So PDQ 39 also good. And scored part 3. This data may be no progression, no progressive using dopaminergic neurons implantation. So we expect IPS derived dopamine neuron also one option for Parkinson's disease treatment. So next speaker, Dr. Hatano, recently advanced in the understanding of development of the Parkinson's disease. So thank you for your attention. I will stop my talk.
Thank you, Professor Hattori. Can you hear me? Or maybe he already out. So maybe time to go the next speaker. Next speaker is Professor Taku Hatano. Could you please start with your presentation?
Okay, Thank you, Professor Tsunemi. Thank you for introducing me and inviting to the nice conference. Professor Hattori mentioned almost part of Parkinson's disease. Audience will understand the almost Parkinson's disease treatment and pathomechanisms. And so I will focus on the alpha-synuclein proteins. And as Professor Hattori mentioned, alpha-synuclein is the most important protein for understanding the Parkinson's disease. This is my COI. Alpha-synuclein might be the culprit in Parkinson's disease because of main component of Lewy body is alpha-synuclein. And alpha-synuclein is relatively small protein, 14 kilodalton. And this protein has two alpha helices. And normal alpha-synuclein can soluble in the water and lipid, but abnormal structure of alpha-synuclein has beta sheet structure. And the beta sheet structures interact with each other and made protofibril and develop fibers. And this is Lewy bodies. And the Lewy bodies contains many fibrillized alpha-synuclein. Furthermore, as mentioned, as Professor Hattori mentioned, alpha-synuclein is a causative gene of autosomal dominant familial Parkinson disease. Until now, the pathogenic mutant was found. Pathogenic mutations was found, has been found, including the V15A, A30P, E46K, H50Q, G51D, A53T. Recently, we identified the V15A is the causative gene of Japanese autosomal dominant familial Parkinson's disease. Furthermore, duplication and triplication of normal alpha-synuclein also cause autosomal dominant familial Parkinson's disease. These findings indicate overexpression of alpha-synuclein will be associated with Parkinson's disease and formed the Lewy body. So alpha-synuclein can bind gently the membrane via the alpha helix structures. This protein is emphatic property, but alpha-synuclein tend to aggregate due to the changes in the binding ability to the membrane. E40K, A53T, H50Q mutant alpha-synuclein tend to aggregate on the membrane, whereas A30P mutant alpha-synuclein decreases ability to bind the membrane. So before two slides, I mentioned Lewy body has many fibrillized alpha-synuclein proteins, but recent CREM analysis can detect the precise structure of alpha-synuclein. This method can match electroscopic histology and can match the electric analysis with immunohistochemistry. So precise Lewy body will be detected. So in this analysis, Lewy body includes a membrane, lysosome, autophagosome, and mitochondria. So this study shows the membrane disruption of alpha-synuclein. A small particle of alpha-synuclein induces aggregation of membranous organelles. So lipid metabolism might play an important role in the mechanism of alpha-synuclein aggregation. So in this lecture, I will focus on the lipid metabolism and Parkinson's disease. First, I would like to talk about the PLH-Z6, which is the causative gene of PARK14. And this protein is a phospholipase. So phospholipid and alpha-synuclein aggregation is very important for the pathomechanisms of Parkinson's disease. And next, I would like to talk about the prosaposin. Professor Hattori mentioned, as Professor Hattori mentioned, we recently found that prosaposin mutation prosaposin mutations cause the autosomal dominant familial Parkinson disease. This protein is key for the glycolipid metabolism. So I would like to move on to the first topic, phospholipid and alpha-synuclein aggregation. PARK14 is caused by the mutations of the PLH-Z6 autosomal recessive disorders, and this patient has homozygous A4990 mutations, and he had adult onset dystonia Parkinsonism, and deep brain stimulation rescued his symptoms. Unfortunately, his sister was died, and autopsy study was performed, and the autopsy revealed that she had many Lewy bodies. PLH-Z6 associated neurodegeneration is known as PLAN. PLAN includes three types of three phenotypes. One is infantile phenotypes, and named as infantile axonal dystrophy. Second is childhood type, known as atypical NAD, and adult is dystonia Parkinsonism, like to him. So we performed to analyze the PLH-Z6 function. So we generated the PLH-Z6 knockout Drosophila model. This figure indicates the motor function. You can see Drosophila knockout model shows the motor dysfunction, and wild type human PLH-Z6 overexpression rescued the motor dysfunction, but AATT mutant PLH-Z6 failed to rescue the motor functions. And this graph indicates the sleep function of Drosophila model. You can see the knockout Drosophila model shows the sleep dysfunction, and wild type overexpression rescues the sleep function, but AATT mutations failed to rescue the sleep dysfunction. And this figure shows the dopaminergic neurons in Drosophila model. Red rectangles indicates the normal Drosophila models, normal Drosophila's dopaminergic neurons, and yellow rectangles indicates the PLH-Z6 knockout Drosophila model. You can see the in knockout Drosophila models, dopaminergic neuronal degeneration was found. And wild type rescues the dopaminergic neurons, but AATT failed to rescue the dopaminergic neurodegeneration in Drosophila PLH-Z6 knockout model. Furthermore, we checked the component of phospholipids, and we found that in knockout Drosophila model, in PLH-Z6 knockout Drosophila model, shows the short acyl chain phospholipid increased. And human PLH-Z6 overexpression can rescue the increased short acyl chain, but PLH-Z6 AATT mutations failed to rescue the increased short acyl chain. And the synaptic, we analyzed the synaptic microstructure. We checked the microstructure of synaptic vesicle in the PLH-Z6 Drosophila knockout models, and this is control. You can see knockout Drosophila model shows the smaller vesicles compared to smaller vesicles than the control Drosophila models. The wild type overexpression PLH-Z6 shows rescue the abnormal structures of vesicles, but AATT failed to rescue. And we also check the physiological function of, we also check the synaptic physiological function. PLH-Z6 knockout model shows decreasing activity. Very interestingly, linoleic acid rescues the PLH-Z6 knockout Drosophila model's phenotype. Linoleic acid decreases short acyl chain phospholipase, and synaptic vesicle structure was normalized, and rescues neuronal degeneration, and rescues synuclein aggregation. This is administration of linoleic acid, and furthermore, rescues motor functions. This is a normal Drosophila and knockout model, and administration of linoleic acid to the knockout Drosophila model. You can see the administration of linoleic acid can rescue the motor dysfunction of PLH-Z6 knockout Drosophila models. So, lipid regulating therapy might be useful for preventing aggregation of alpha-synuclein. So, what you eat can affect your risk of developing Parkinson's disease. A balanced diet might prevent Parkinson's disease. Several epidemiological studies revealed that Mediterranean diet might reduce the risk of Parkinson's disease. So I would like to move on to the next topic, glycolipid and rhizosome. Glycolipid metabolites in the glycolipid was metabolized in the lysosomes, and the knockout of the glycolipid enzymes showed the lipidosis. Lipidosis means aggregation of abnormal glycolipids. So glucocerebrosidase is one of the most famous lipidoses, and double knockout of glucocerebrosidase induce Gaucher disease, but heterozygote GBA mutation is a highest risk gene of Parkinson's disease. Similar to the glucocerebrosidase, SMPD1 is known as Niemann-Pick type A and B. SMPD1 is also a heterozygote SMPD1 PL302P mutation is a risk gene of Parkinson's disease. Allyl sulfatase A, which is known as metachromatic leukodystrophy. Heterozygote alpha allyl sulfatase A is a risk gene of Parkinson's disease. This paper was published by my colleagues and, of course, Professor Hattori. So, so we focused on the sapochins. Sapochins are activators of glycolipid enzymes. Sapochin assists the function of glycolipid enzymes in lysosomes. These proteins are originated from prosapocin. Prosapocin was traveled, prosapocin is traveled from Golgi to Golgi to the lysosomes. In the lysosomes, prosapocin is degraded by the catepsin D and divided to 4 sapochins. And each sapochin co-activates its corresponding enzymes. For example, sapocin A collaborate with galactosylceramidase, sapocin B collaborate with allyl sulfatase A, sapocin C collaborate with glucocerebrosidase, and sapocin D collaborates with acid ceramidase. And deficiency of each sapocin causes a disease phenotype mimicking lipidosis due to the deficiency of the lysosomal enzyme. Deficiency of sapocin C induce glucose velocidase mimic lipidoses. So we found saposin D mutations are associated with autosomal dominant familial Parkinson's disease. We found independent 3 families, and they have different mutations. Family A has Q453P mutation. Family B has a deletion, and family C has C412Y mutation. And this structure is saposin structure, and this is saposin D, and saposin has 3 SS bonds, and these SS bonds is important for the function of saposin, and all mutations affect the SS bonds. And the family member shows decreasing MIBZ cardiac scintigraphy, and that scan shows nigrostriatal dysfunction of nigrostriatal pathways and indicates dopaminergic neurodegeneration. And this finding is quite similar to the sporadic Parkinson's disease. And furthermore, saposin D domain mutations affect the prosaposin trafficking. And Lefrancois and Colliers reported that the prosaposin D domains bind to sphingolipid to function as prosaposin trafficking. And this is a control IPS, and these are the control IPS, and these are the IPS derived from the patient, and the Q453P deletions and C412Y. And the red indicates the prosaposin, and green indicates the PDI. PDI means PDI is ER marker. And you can see that in control subject, prosaposin did not colocalize with PDI, whereas mutations, mutant in the mutant iPS cells, prosaposin colocalize with ER. So pathogenic mutant prosaposin shows the impairment of its trafficking to lysosomes. Furthermore, we investigated the fibroblast from the control and pathogenic mutant patients, and this structure indicates autolysosome. You can see the controls compared with control subject. Pathogenic mutant families, pathogenic mutant patient has abnormal autolysosomes. And furthermore, overexpression of LAMP2 was found. LAMP2 is known as lysosome marker. So patient's fibroblast shows abnormal lysosomes. Furthermore, we investigated the mouse model. We had a mouse with C509S prosaposin mutations. This mutation is quite similar to the family C mutations. Red indicates heterozygote mouse model, and blue indicates homozygous mouse models, and this is motor function. You can see the heterozygote mutations and homozygote mutations is showed the motor dysfunction like to the patients. And furthermore, we check the dopaminergic neurodegeneration in this mouse model, in this mouse model. And heterozygote and homozygote mutant mice had neuronal degeneration of dopaminergic neurons. After publishing our data, Chinese group also reported Parkinson's disease patient with mutation of prosaposin. And this patient has E45QD. This is saposin D domain. This mutation locates in the saposin domain. And the saposin A, B, C also mutations in the saposin A, B, C also cause autosomal dominant familial Parkinson's disease. I would like to move on to the final topics, propagation of alpha-synuclein. As Professor Hattori mentioned, fibrillized alpha-synuclein inoculation of mice brain shows the propagation of alpha-synuclein aggregation. And red dots indicates aggregation of alpha-synuclein after 6 months after inoculation of the fibrillized alpha-synuclein in the striatum. So we check the transmission manner of alpha-synuclein aggregation. And we check the retrograde marker and anterograde marker. Retrograde marker includes the NFH and TH, and the anterograde marker beta 4 and NAV 1.2. And green indicates alpha-synuclein aggregations and propagation. And red indicates the retrograde marker. You can see the alpha-synuclein merged with the retrograde marker. And also, alpha-synuclein merged with the anterograde manner. So this cartoon explains my explained our examination. And this is cortex, and this is putamen, and this is corpus callosum. And the neurons connect like this, and inoculation of alpha-synuclein seeds propagate via both the retrograde and anterograde manner. Next, we perform the callosotomy. Cutting the neurons will prevent the alpha-synuclein transmission. So we performed 24 hours after and before alpha-synuclein inoculation into the contralateral side of striatum. So red rectangle indicates the 24 hours pre-callosotomy, and blue rectangle indicates post-24 hours post-24 hours after alpha-synuclein inoculation. You can see post-24 hours callosotomy cannot prevent the propagation of alpha-synuclein. On the other hand, pre-callosotomy can prevent the propagation of alpha-synuclein. So these findings indicates alpha-synuclein propagation in experimental model is very, very rapid manner. So next, we asked, what is the mechanism of propagation? Assumed propagation mechanisms include exocytosis, endocytosis, exosomes, tunneling nanotube. So to this end, we employed botulinum toxin. Botulinum toxin cleaves snare protein, resulting in the prevention of exocytosis. We performed whether botulinum toxin will prevent the transmission of alpha-synuclein. So we performed the alpha-synuclein inoculation and before 1 day before contralateral side botulinum toxin. One day after the botulinum toxin treatment, and before 3 days botulinum toxin treatment. This method indicates alpha-synuclein inoculated to the botulinum toxin. This method indicates a pre-treated botulinum toxin. Red rectangle indicates the pre-treatment of botulinum toxin. Pre-treated botulinum toxins can prevent the propagation of alpha-synuclein. So this is my talk's conclusions. A Lewy body includes lipid membranes, lysosome, autophagosomes, and mitochondria. Dysfunction of PLX-D6 induces a disturbance of lipid composition resulting in neurodegeneration with alpha-synuclein aggregation. Dysregulation of glycolipids metabolic pathway plays an important role in the pathomechanisms of Parkinson's disease. Prevention of alpha-synuclein propagation might be one of the targets for the disease-modifying treatment of Parkinson's disease. They are collaborators. That's all I have to say. Thank you for your attention.
Thank you, Professor Hatano, for you're covering from the Levy bodies to alpha-synuclein propagation. Thank you for a nice talk. So we move on the next speaker, Dr. Saiki. Could you please start with your talk?
Thank you. Thank you for giving this wonderful opportunity presenting with our data associated with Parkinson's disease biomarkers. The title is Novel Biomarkers for Detection of Early PD, Parkinson's disease. I would like to share. Okay. I would like to show two topics in this presentation: polyamic metabolic changes in Parkinson's disease and another topic is accumulation effects on autophagy via lysosomal accumulation. For 10 years, we have been researching on Parkinson's disease biomarkers using biological specimens, especially blood. These schematic figures showing the association of disease progression with percent of residual nuclear dopamine neurons, this axis, and x-axis mean times, y-axis does percent of residual substantial nuclear dopaminergic neurons. In the clinical course of Parkinson's disease onset occurs in this point, onset of neurodegeneration. And dopaminergic neurons gradually decreases up to 50%. They show apparent motor symptoms, so-called symptomatic phase. Importantly, the neurodegeneration initiated at least more than 10 years ago. Previously, we have reported various kinds of PD biomarkers like that. In this presentation, I would like to focus on these 2 points: polyamic changes in Parkinson's disease and accumulation increase in PD. Both of them are assayed with serum. Polyamic mainly includes psoracin, spermidine, and spermine, which are polyvalent phytochemicals contained in soybeans, fungi. Feature polyvalent phytochemicals contained in soybeans, fungi, and natto, a kind of Japanese traditional and popular fermented food. Why do we research on polyamic effects on neurodegeneration? As you know, the highest risk of Alzheimer's disease and Parkinson's disease is aging. You can see the gradual increase of the disease prevalence, AD and PD respectively. The prevalence of AD is around 10% at age 80, also 1% of Parkinson's disease. Polyamines, spermidine and spermine induced longevity by autophagy activation via various protective mechanisms, including anti-cancer, cardioprotection, neuroprotection, anti-inflammation, and effects on stem cells. Previously, we have shown that colitis is repressed by polyamine enhancement in mice. Okay, First of all, I show the patient characteristics of the cohort study. The numbers of controls and PD patients are 4D9 and 186. No significant differences between them were detected except for cognitive function. This is a summary of the results. In polyamic metabolism, ptorescin is converted to spermidine, which is converted to spermine consecutively. Oh, just a moment. And the levels of G-acetylspermidine gradually increases with disease severity like that. Also, spermine levels are decreased, and first, those of spermidine increased in Parkinson's disease. So, the ratio of spermine to spermidine is continuously decreased in Parkinson's disease. However, the ratio is gradually decreased in controlled patients. This slide shows the serum G-acetylspermidine level is a brain translator biomarker. The x-axis means levels of G-acetylspermidine, and y-axis means functional anisotrophy with reflection of axonal dysfunction of the white matter. You can see the apparent positive correlation between them. Importantly, lesions with high fractional anisotrophies are detected diffusely in the white matter of Parkinson's disease patients, colored by yellow and red, implying that you can access the brain abnormalities only by blood sampling with analysis of G-acetylspermidine. Okay, next topics. Let me turn to the next topics. Next, we seek for the mechanisms of the effects of spermine as well as accumulation on autophagy in neuronal cells. These pictures show the green dot distribution change in neuronal cell lines. You can see the accumulation of the green dot by treatment of spermine, N1-acetylspermine, and spermidine like that. Lysosomes around the black circle, around the blue circles, but other chemicals show the scattering distribution of the lysosomes. This schematic figure shows autophagy regulation via lysosomal distribution change. Lysosome nears cell membrane continuously receive peripheral upstream signaling toward mTORC1 complex, resulting in inhibition of autophagy. However, lysosomes near the nucleus are unable to collect mTORC1 stimulation from the cell membrane, leading to autophagy activation, and are easily fused with lysosomes around the nucleus. Those result in enhancement of autophagy flux. Thus, we checked accumulation effects on neuronal cell lines. You can see the marked increased levels of serum accumulation in Parkinson's disease compared to controls. Also, lysosomal accumulation as well as autophagy upregulation were identified by immunocytochemistry as well as Western blotting. Lysosomal transport to the nucleus are organized by various dynein complexes like that. Next, we investigated that gene silencing against each component of dynein complex, GIP4 or TMEM, 55B, ALLG2, TRPML1, and RAB7. You can see the marked accumulation of the lysosomes around the nucleus by controlled siRNA. However, knockdown against ALG2, TRPML1, and GIP4 induced the lysosomal distribution change to the scatter patterns compared to the control or RAB7 or TMEM55B. Next, we investigated them with TRPML1 regulates calcium efflux from the lysosomes like that. So we investigated them with immunofluorescent techniques. You can see the calcium efflux increase by acrolein treatment like that. The consecutive event followed by calcium efflux enhancement is expected to induce calcium-dependent kinase activation. You can see the blocks of lysosomal accumulation by calmodulin kinase 2G knockdown like that. But against CAMK2A, CAMK2B, or CAMK2D knockdown did not change the localization of the accumulated lysosomes. We noticed that acrolein induces hyperphosphorylation of GIP4 by normal Western blotting techniques. Thus, using host-tag page techniques, we can confirm the GIP4 hyperphosphorylation by acrolein treatment like that. The band is shifted to the upward. Also, we confirmed that upstream associated CAMK2G was also inhibited by JAK3 inhibitor 4 like that. So the band shift by acrolein treatment is blocked by JAK3 inhibitor like that. Next, we identified phosphorylation site of GIP4 in T217 by acrolein administration using mass spectrometry. You can see the marked increased levels of phosphorylation at T217 like that. Also, those hyperphosphorylations are prohibited by acrolein plus JAK3 inhibitors treatment. Next, we performed immunoprecipitation experiments. Those experiments revealed that interaction of TRPML1 and GFP-ALG2 like that. So, acrolein treatment enhances the binding between them, and dynein complex functions are also enhanced by acrolein. Next, we reconfirmed that this lysosomal positioning regulation by oxidative stress is different from that of starvation-induced autophagy, because acrolein is toxic and induced oxidative stress like H2O2 treatment. You can see the marked accumulation of the lysosomes around the nucleus in control cells treated by DMSO like that. However, oxidative stress, those were blocked by BAP-TA and JAK3 inhibitor only in oxidative stress status. The GIP4 phosphorylation which is a critical step of the system, is enhanced by H2O2 treatment as well as acrolein treatment like that. requires that was not observed by starvation conditions. As shown in this upper graph, the oxidative stress increases the cell death because of its severe toxicity like that. If we weaken the function of the system by GIP4 knockdown or TRPML1 knockdown, you can see the significant enhancement of the cell death assayed by LDH assay. So we concluded that this lysosomal accumulation changes induced by H2O2 treatment as well as Accumulation treatment is neuroprotective effects of the regulated by phosphorylated GIP4. This is a schematic summary of our study. Serum acrolein levels are elevated in Parkinson's disease and would be a potential biomarker of Parkinson's disease. Also, acrolein induces calcium efflux from the lysosomes, and calcium efflux activates the calmodulin kinase 2D, 2G, and GIP4 is hyperphosphorylated. Hyperphosphorylated GIP4 enhances the binding of the TRPML1 and ALG2 and dynein complexes, resulting in accumulation of the lysosomes around the MTOC, microtubule organizing center. This autophagy alleviates oxidative stress or cytotoxicity. This novel mechanism is not observed in starvation-induced autophagy. This is a summary of my presentation. N18G-acetylspermidine is a brain translatable biomarker for Parkinson's disease. Spermine acrolein induces autophagy via lysosomal accumulation around the microtubule organizing center or nucleus. Oxidative stress-induced phosphorylation of GIP4 regulates lysosomal positioning in coordination with TRPML1 and ALG2. Thank you for your attention.
Thank you, Professor Saiki, for your nice talk. And so we're gonna move on the next one. Actually, next one is my turn. So I'm gonna speak about the exosomes. So I will talk about the extracellular vesicles, which are now considered important in Parkinson's disease. Our body fluid contains a lot of extracellular vesicles. Extracellular vesicles can be divided into two categories, ectosomes and exosomes. Ectosomes are vesicles that pinch off the surface of plasma membrane via outward budding and include microvesicles, microparticles, and large vessels in the size range of 50 nanometer to 1 micrometer in diameter. Exosomes are extracellular vesicles with a size range of 50 to 150 nanometer with an endosomal origin. Sequential invagination of the plasma membrane ultimately results in the formation of multivesicular bodies, which can interact with other intracellular vesicles and organelles, contributing to diversity in the constituent exosomes. Depends on the cell origin, extracellular vesicles, including exosome, can contain many materials of a cell, including DNA, RNA, lipids, metabolites, and cytosolic and cell surface proteins. The physiological purpose of generating exosomes remains largely unknown. One speculated role is that exosomes likely remove excess or unnecessary materials from the cells to maintain cellular homeostasis. Recent studies indicate a functional, targeted, mechanism-driven accumulation of specific cellular components in exosomes, suggesting that they have a role in regulating intracellular communications. As shown in the bottom figure, extracellular vesicles attract much attention in the scientific fields. Exosomes are mediator of near and long-distance intracellular communication and affect various aspects of cell biology. This diagram shows the involvement of exosomes in multiple cellular functions: gene regulation, proliferation, reproduction. Recent study revealed that involvement of exosomes in various disorders, one of which is Parkinson's disease. I will focus on exosomes' function in waste management. Exosomes are generated within the multivesicular bodies over endosomal lysosomal pathways. Genes that are implicated in familial forms of PD, such as VPS35, ATP38, LAC2, and Parkin, function in the pathway as shown in the diagram. Alpha-synuclein misfolded is considered as a key pathogenic event in sporadic PD. Alpha-synuclein is tracked via endosomal pathway and released in exosomes. Exosomal alpha-synuclein, LAC2, or other proteins may contribute to PD pathology by modulating microglial cell activation, facilitating the propagation of alpha-synuclein. I have been working on exosomes and its relation to Parkinson's disease. I will share my past findings. First, exosomes contribute to alpha-synuclein accumulation in neurons. Second, exosomes can be used as a biomarker for Parkinson's disease. Third, exosomes are involved in the propagation of alpha-synuclein. Let me start with brief description of Kupffer-Lakev syndrome. This is a rare autosomal recessive neurological disorder. The patients show juvenile or early onset of Parkinson's disease symptoms. The earliest age of onset is 6 years old, and the average age of onset is 22. Importantly, the treatment of L-DOPA is effective to Parkinsonism, suggesting that dopaminergic neurons in substantia nigra are affected. That is why Kupffer-Lakev syndrome is categorized in one of the monogenic forms of Parkinson's disease. However, the clinical symptoms of disease are not only what we see Parkinson's disease patients. These patients show supranuclear gauge palsy and spasticity and dementia. Data scan shows reduced dopaminergic transport activities in striatum, and brain MRI shows mild atrophy, and some patients suggest an accumulation of iron. So far, 11 families and 37 individuals have been reported. 2006, the gene was discovered, and it is ATP38 that causes type 5 P-type ATPs. From amino acid sequence, it is predicted to serve as a cation transporter. It also transports polyamines. We and others have reported that PARK9 loss of function leads to lysosomal dysfunction and increased alpha-synuclein expression levels. ATP38 mutations cause not only Kupffer-Lakev syndrome, but also typical PD and familial spastic paraplegia. The alpha-synuclein expression levels are closely associated with Parkinson's disease. Therefore, its regulation level is critical for normal neurological function, and there are multiple degradation pathways: ubiquitin proteasome pathway, chaperone-mediated autophagy, the macro-autophagy, and endocytotic pathway. These pathways finally converge to lysosome. Recent studies suggested that alpha-synuclein proteins have a secretory future. We found that ATP38 Park9 located in both lysosomes and multivesicular endosomes, and reported that impaired intraluminal vesicle formation contributed to reduced exosomes and alpha-synuclein release. While Park9 has been shown to localize acidic vesicles, the nature of these vesicles has not been elucidated in detail. To further examine Park9 localization, we expressed Park9 V5 expression plasmid in H4 cells and conducted double immunostaining. Cascade blue conjugated dextran was used for late endosome and lysosomal markers. Park9 co-localized with LAMP1 positive vesicle suggested that localization to late endosome and lysosome using antibody against CD63, a more specific marker for multivesicular region of endosome, we found that Park9 also co-localized with multivesicular endosomes. To confirm these findings, immuno-EM with antibody against V5 Park9 and CD63 was performed. A few Park9 V5 labeled gold particles were associated with CD63 vesicles, suggesting that Park9 co-localized, at least in part, to multivesicular bodies, possibly to the outer membrane of the vesicles. To further analyze these multivesicular endosomes, we performed EM analysis with wild-type and Park9 mutant fibroblasts. We found that reverse-packed intraluminal vesicles in multivesicular bodies is wild-type fibroblasts, whereas interluminal vesicles were very sparse and very few in Park9 mutant cells, suggesting that Park9 may control the number of interluminal vesicles in multiple vesicular bodies. Since Park9 mutant cells contain fewer interluminal vesicles, we asked whether the number of exosomes is affected by Park9 mutations. After loading exosomes by conventional centrifuge, we performed Western blotting using established biomarker for the exosomes ARIX, protein 1, TSG101, and CD63 to confirm the identity of collected exosomes. These results suggest that exosomes from Park9 mutant fibroblasts were fewer compared to wild-type fibroblasts. Then we further analyzed exosomes by nanoparticle tracking analysis. This way, we can visualize the exosomes to simultaneously quantify the size and the number of exosomes. Park9 mutant fibroblasts released small but significantly lower number of exosomes compared to wild-type fibroblasts. These results were consistent with the finding of fewer intraluminal vesicles in multiple vesicular bodies. Next, we hypothesized that Park9 may be involved in the secretion of exosomes. In support of this hypothesis, we found that overexpression of Park9 led to increased number of exosomes compared to vector-infected controls. This result was confirmed by Western blot analysis using exosome marker. Taken together, these findings further support the notion that Park9 play a role in the production of exosomes. Recent evidence suggests that alpha-synuclein is at least in part secreted from neurons via exosomes. Since our findings suggest that Park9 regulated exosome release, and the fact that alpha-synuclein accumulate in cells, animal models of Kupffer-Leber syndrome, we analyzed alpha-synuclein secretion with a highly sensitive ELISA. First, we confirmed our previous data, and that silencing Park9 with shRNA significantly increased alpha-synuclein protein levels, whereas overexpression of Park9 reduces the level of alpha-synuclein. We confirm these findings by ICC. Park9 overexpression decreased alpha-synuclein expression. Then we analyzed alpha-synuclein secretion in media and exosome and found that Park9 silencing reduced and Park9 overexpression increased alpha-synuclein in both media and exosomes. These results suggest that possibility that Park9 regulate alpha-synuclein levels inside the cells by secreting alpha-synuclein by exosomes. We found that loss of Park9 leads to impaired inter-element vesicle formation, contributed to reduced exosomes and alpha-synuclein release. Also, there is an inverse correlation between alpha-synuclein levels in neurons and exosomal alpha-synuclein. So second, our finding is exosomes can be used as a biomarker for Parkinson's disease. To this end, we isolated exosomes from serum and analyzed the number of exosomes and exosomal alpha-synuclein. Exosomes are quantified by nanoparticle tracking systems, followed by purification and from serum by conventional ultracentrifuge. The isolated exosomes is positive for the marker or exosomes mark. EM analysis also shows the typical exosome forms. We also developed ELISA system for quantification of filamentous alpha-synuclein. Clinical characteristics of the patient. We have collected from 105 controlled people, 142 PD patients, 18 MSA patients, multiple system atrophy patients, and 28 progressive supranuclear palsy patients. The amount of exosome significantly decreased in PD compared to both control and atypical Parkinson's. In contrast, the filamental alpha-synuclein in exosome significantly increased in PD compared to both controls and atypical Parkinson's, suggesting that exosomes and the alpha-synucleins in exosome can be a biomarker for the Parkinson's disease. My third evidence is exosomes are involved in the propagation of alpha-synuclein. The alpha-synuclein travels from cell to cell in multiple ways. Recent evidence also suggests that alpha-synuclein, a small protein with unknown function, may spread in prion-like manner, but the underlying mechanism of the propagation remained undetermined. The alpha-synuclein traveled from cell to cell in multiple ways. Initially, alpha-synuclein was transmitted by synaptically. However, results from animal model revealed its limitation. The alpha-synuclein moved from cell to cell in a tunnel nanotube, but not all cells. The alpha-synuclein is secreted in multiple pathways, including exosomes, that's what I talked, autophagic secretion, and lysosomal degradation. The secreted alpha-synuclein are taken up by other cells. Initially, most researchers hypothesized transneural transmission. Indeed, single inoculated of alpha-synuclein preformed fibrils, PFS, installated resulted in Lewy pathology, predominantly anatomically connected lesions, in a time-dependent manner. However, if we look carefully, it already spread in entire brain at early time point. Single inoculated PFFs in the olfactory bulb spread it to the region located 2 synaptic away, but it took for 360 days, and then they stopped spreading. These results reveal the limitation of transneuron spreading and suggest that existing of alternative pathway, which can spread alpha-synuclein in the entire brain. One of the candidate is exosomes. In addition, exosomes secreted from microglia play a significant role in alpha-synuclein propagation. PD patient-derived exosomes can activate microglia. Pharmacological depletion of microglia by PLX33-19-7 suppressed alpha-synuclein propagation in mouse brains. Alpha-synuclein is uptaken by microglia through TLR2 receptors. These evidence suggests the involvement of microglia in alpha-synuclein propagation through exosomes. As I have shown you, we have reported that ATP38 to PARC9 regulate exosome release and determines intracellular alpha-synuclein in H4 and iPS cell-derived DA neurons, suggesting that exosomes release can be changed by ATP38. We set to examine the role of exosomes in alpha-synuclein spreading in the brain. To this end, we used ATP38 to neuromines where exosome secretion is decreased. Sonicated PFFs were injected in the right striatum of the mice ranging between 2 to 3 months of age. At 3 months after of injection, the mice were fixed and immunohistochemical analysis were performed with an antibody, alpha-synuclein antibody. The distribution of Levy bodies, red circles, and Levy neurite, red line, like inclusion in the wild type and ATP38 to neuromines at 3 months after mouse PFS inoculation. The representative image of each section from the wild type and ATP38 to neuromines. The results of quantitative analysis, Levy bodies, and Levy neurite-like inclusion in each section shows that there is no difference between wild type and ATP38 to neuromines, suggesting reduction of exosome does not affect alpha-synuclein propagation. Then we analyze the effect of enhanced exosome secretion on the propagation of alpha-synuclein. To this end, prior to injection of sonicated PFFs, ATP38 expressing lentivirus were injected in the same position. The distribution of Levy bodies and Levy neurite-like inclusions in the wild type ATP38 and ATP38 neuromines, where ATP38 was overexpressed at 3 months after mouse PFS inoculation. The representative image of each section from the wild type and ATP38 neuromines, where ATP38 was overexpressed at 3 months after mouse PFS inoculation. The result of quantitative analysis of Levy bodies and Levy neurite-like inclusions in each section shows that Levy bodies increased around the injection site, but they decreased at the regions far away from the injection site, suggesting that enhanced exosome secretion decreases alpha-synuclein propagation. So it's my conclusions. First, exosomes contribute alpha-synuclein accumulation in neurons. Yes, the number of exosomes and the intracellular alpha-synuclein levels are inversely correlated. Second, exosomes can be used as a biomarker for Parkinson's disease. Yes, exosomes can be a promising biomarker for Parkinson. Third, exosomes are involved in the propagation of alpha-synuclein. Yes, but surprisingly, enhanced exosomal secretion reduced alpha-synuclein propagation. Thank you for your attention. So my talk is over. So now we have four speakers, and all speakers is now finished speaking. Yeah, but Taku, so we don't have any question from the audience. But we have 30 minutes. We can talk. Or we can close the workshop. Professor Hatano, do you have any questions or anything to add topics on this workshop?
Okay. This workshop is very wonderful, and all speakers very talked about the nice topics. So we should discuss with each other, and I have a question for the Dr. Tsunemi's presentation. You mentioned the exosome will have the important for the aggregation and propagation of alpha-synuclein. And the recent research reported that derived from neuronal exosomes contains very small particle of the alpha-synuclein oligomer. Do you think neuronal derived exosome is the most important exosomes, or other derived other exosomes from exosomes from other organelles, for example, the red blood cells and white blood cells and other and so on, is I think is also important for the propagation of alpha-synuclein. Do you think our specificity of the exosomes and alpha-synuclein aggregation will be important? Do you think?
Actually, this is a nice point you said. We have believed exosomes are very important secretory pathway of alpha-synuclein, which determines the levels of intracellular alpha-synuclein. But as you mentioned, there is a lot of other secretory pathway, exosomes, microvesicles, and also actually that what I published before, lysosomal exocytome. All these secretory pathways can release alpha-synuclein and then contribute to alpha-synuclein levels in the cells. And then I found that the number of exosomes released from the neurons actually much smaller, maybe 10 times or maybe 20 times less than other type of cells, especially in glia. So maybe as you said, neuronal exosome contribution to the intercellular alpha-synuclein levels in neurons, maybe not so much, but that's what I expected. But continuously, releasing of exosomes, maybe some play a role to determine the intraneural alpha-synuclein levels. But if you talk about the glia's exosomes, there's 10 times more exosomes. And then recent studies suggest that if you stop the, for example, microglia secretion of the exosome, you can reduce the spreading of alpha-synuclein in mouse brains. Suggests that when you talk about the spreading of alpha-synuclein, maybe glia's exosomes contribute much more.
Thank you. And I think red blood cell and platelet contains many alpha-synuclein proteins. And in addition, the inflammation is also important for the neurodegeneration in Parkinson's disease. So I'm very interested in the white blood cells and reticular cells, maybe reticular cells contains many alpha-synucleins, maybe trigger the alpha-synuclein aggregation in peripheral nerves, I think. So very...
Yeah, I think so. I think it's also related to the development of technology because when I started working on the Parkinson's disease, it's just probably less than 10 years ago. At that time, people said alpha-synuclein predominantly or exclusively located synapse in neurons.
Yeah.
But now, so we have a very high sensitive ELISA or other ways, other technology, which can detect a very small amount of exosomes that can reveal now, alpha-synuclein is everywhere. And then very small but the toxic alpha-synuclein can be, matter more in Parkinson's disease. So I think we are right. So like a white blood cell or red blood cell, they also contain alpha-synuclein, and maybe that affect Parkinson's disease pathology.
Thank you so much. And I have also one question to the Dr. Saiki, Professor Saiki. You mentioned accumulation is important for the autophagy systems. And recent, I mentioned Lewy body will be contains disruption of the several membrane organelles. Do you think the accumulation may associate with forming the Lewy bodies? Do you have any comments?
Thank you for your question, Dr. Hatano. It's quite a difficult question because accumulation is elevated in blood, in serum. So polyamic metabolism is regulated in all kinds of organs in the whole body. So I'm not sure the exact function of the polyamic, including accumulation, affects on specific protein aggregation, especially alpha-synuclein. So the multifunctions of polyamic metabolites or polyamic itself is quite complex. So I wish it affects on the protein aggregation or protein clearance of specific alpha-synuclein. But it's quite tough question for me in the present data.
So in addition, recently the microbiome may be important for the pathomechanisms of Parkinson's disease. And the microbiome make many metabolites in gut. So your metabolic analysis found several metabolites for the association with Parkinson's disease. Do you think the correlation between the your found metabolites and the microbiomes in Parkinson's disease?
Thank you for your question. That's a very important point, I think. In terms of our metabolites detected in previous studies, the polyamine metabolites would be produced by microbiome. So regulation of microbiome in Parkinson's disease patients would be useful for treatment because it's focusing on polyamine metabolism. However, recent studies have shown that the importance of short-chain fatty acid is well established in Parkinson's disease because those kind of fatty acids are protective, definitely, to regulate colon inflammation. So it might be important, but polyamine metabolites should be regulated by microbiome, but we should seek for the correlation or correlation between microbiomes, colon microbiomes, and serum metabolites.
Yeah.
So those kind of studies should be performed.
Yeah. I think it's a very tough work, but very important, I think.
So I have a question for Professor Hatano.
Yeah.
So, alpha-synuclein propagation theory is now very popular in research field and the clinical field.
Yeah.
But do you think this, a lot of experimental results, alpha-synuclein once inoculated in gut or brains spread to, other parts of the brain or bodies? Is this really happened to human patients? What do you think?
Very important questions. Inoculation mouse model shows a very, very quiet, quick propagation of alpha-synuclein. It's not reflect the human pathology, I think, because Parkinson's disease patients shows a very, very slow progression of disease. So the inoculation, alpha-synuclein aggregation and propagation theory may be in experimental models, restricted in the experimental models, but pathological findings of the Parkinson's disease, around the 60% patients shows can be explained by the propagation theory. Olfactory bulb and vagus nerves is the first step of the aggregation of alpha-synuclein. So several patients may be explained by the propagation theory, but for example, dementia with Lewy bodies, and some patients shows a fulminant Lewy body disease, shows very rapidly and widely propagation of alpha-synuclein. So the findings cannot be explained by the only transmission from the neuron to neuron. It's difficult to explain, I think. So I think one of the propagation and transmission from neuron to neuron will be the pathogenesis of Parkinson's disease, but the other pathway will be important for the pathomechanisms of Parkinson's disease, I think.
Thank you. What do you think? I would ask the same question to Dr. Saiki.
It's a very important, but
Professor
Hattori.
Hattori raising his hand.
How about
this is-
yeah?
So I think Dr. Hatano talked about quick propagation of alpha-synuclein protein. So this is the exact discrepancy between experimental propagation and clinical progressive course. So this means that I think the host factor could be involved in onset of Parkinson's disease, including immunological system, also one of the defense mechanism for propagation. So therefore, we have to see the true mechanism between defense and propagation system. And also exosome, this is very important. Exosome easily pass to BBB, blood-brain barrier. And also, I'm not sure synuclein fibrillate inside of exosome or attachment of exosome membrane. So this is also another question for propagation system. Now, recently, so the black dual hit hypothesis is very, very fascinating and for us. But not only dual hit hypothesis could not explain skin deposit of alpha-synuclein and other organisms. So this is another maybe multiple pathway for propagation system could be involved in Parkinson's disease progression. This is my comment.
Thank you. It's very interesting, very hot field for the research field. But actually, I'm always little bit hesitated to all these our finding can be transmitted to the human patient or not. So Professor Hattori, I have another question. You show there is a 24 or maybe 25 monogenic forms of Parkinson's disease.
Yeah.
And then those genes are roughly divided to pathways.
Yeah.
One is a mitochondria and one is a lysosomal pathway. So our next step is how to convergent, how to find a final answer, or this one is just we have a two pathway, or maybe there is a one pathway which can explain both ways. What do you think?
I think in Parkinson's disease, if we have 10 Parkinson's disease, such patients have 10 different pathomechanisms of Parkinson's disease. And one of them is mitochondria and inflammation, lysosome, and autophagy impairment. So a monogenic familial Parkinson's disease provides us good hint to elucidate the pathogenesis of this disease. For example, some Parkinson's disease primary mitochondrial dysfunction. Other lysosome, the other lysosome, or other lysosomal impairment, or autophagy impairment. So in the near future, we have to bank of IPS of each patient. A patient show mitochondrial dysfunction. B patient show lysosomal dysfunction, including autophagy, rhizosomal dysfunction. So this means each different pathway could be common mechanism. Common mechanism only we know dopaminergic neuron could be susceptible for each mechanism. Only one mechanism means dopamine deficiency could be involved in dopamine mechanism. Only one pathway may be common pathway for various monogenic familial Parkinson's disease, including sporadic Parkinson's disease. Do you understand?
And I have also one comment, and I agree with Professor Hattori's answer. And in addition, I mentioned claim analysis reveals that almost three bodies contains many organelles, including the mitochondria, lysosome, autophagosome, and synaptic vesicles. So in Parkinson's disease, all no pathways found from the monogenic Parkinson's disease will be important for the pathomechanisms of Parkinson's disease, mitochondrial dysfunction and lysosomal dysfunction, the synaptic moving dysfunctions. All of them is associated with Parkinson's disease, neuronal degeneration. So it's very complex. So we have to do the many things. I think so. It's my comment.
Thank you. So it's almost the time is up, but I would ask all the three speakers one last question. What do we have to do next? What is the next most important question, the important steps for the Parkinson's disease for cure the patient? What do you think, Professor Hatano?
Yeah, I'm very interested in the aggregation of alpha-synuclein and how to form the Lewy bodies. So I focused on the how to synuclein aggregations. Why the synuclein aggregations? What is important for the triggers of alpha-synuclein aggregation? I hope to reveal these issues. Thank you.
Thank you. How about Dr. Saiki?
Mute.
Mute.
So mute, please.
Please, mic on.
Mic
on.
Thank you. The most important strategy is intervention against alpha-synuclein ASAP, especially in prodromal or preclinical phase, I think.
Thank you. Professor Hattori, could you conclude? What do you think the most important next step for the Parkinson?
So thank you so much. So now there is no, there is currently no cure therapy for Parkinson's disease. So therefore, the next step, we have to seek for disease-modifying therapy. So disease-modifying therapy may hold the progression of this disease. And then, so we have to see true mechanism of synuclein aggregation. And so there is no cure therapy, and therefore the later on cure PD. So therefore the next step, disease-modifying therapy. Maybe to stop synuclein aggregation or synuclein propagation. This is the final target. Our group have to so targeted from true mechanism of Parkinson's disease. This is my conclusion.
Thank you very much. So our workshop titled Parkinson's Disease from Pathophysiology to Treatment is now over. We hope that our presentation helped your understanding of this intractable disorder and paves the way for the development of fundamental treatment in the near future. Thank you for participation. Bye-bye.